Miriam, of course, is well known to the Barth community for a number of years. She's been involved with tafazin knockdown yeast, developing that model, and also involved with CRL or CRD mutants, cardiolipin synthase mutants in yeast, and there are a lot of studies. Title is right here: Loss of cardiolipin leads to perturbation of mitochondrial and cellular iron homeostasis. Miriam.
Thanks, Grant. I'd like to start by thanking the organizers for giving me this opportunity to talk about our work, and also, of course, thanking the Barth Syndrome Foundation for putting together this really amazing meeting. It's always, I'm always overwhelmed at how fantastic the meeting runs, and it's so complicated, and they just, we make it happen. You guys make it happen, so thank you very much. In my laboratory, we've been interested in understanding the functions of cardiolipin for many years. And we have determined that cardiolipin plays an important role in many essential cellular functions. What I'd like to talk about today is the role of cardiolipin particularly in iron homeostasis. And there will be echoes of Richard Kelly's talk towards the end, as we'll hopefully talk about how this is hopefully relevant to Barth syndrome. So our understanding of Barth syndrome has had the trajectory of first with Peter Barth identifying the disorder in 1983, and the identification of the gene in 1996, quite a number of years later. And then the really important finding by Peter Freken and his co-workers, Ron Vonder's group, in 2000, that the biochemical defect has to do with cardiolipin remodeling, and in particular, and also relevant to what I'll be talking about, the biochemical defects are decreased cardiolipin, increased monolysocardiolipin, and decreased unsaturated fatty acyl species. Now, I think it's important to note that we don't know which of these biochemical defects really is the cause of the disorder. So that it's important to keep that in mind. And then continuing along with the biochemical defect, the really pivotal paper of Michael Schlama showing that there's a deficiency of L4 cardiolipin in Barth syndrome. And so I think that where we are now is trying to understand the molecular basis of the pathology and also the relationship of genotype to phenotype, because as we know, there's a huge disparity in symptoms among patients of Barth syndrome, even with patients that have the identical mutation. So this raises the question of why is it so hard to understand the pathology of a simple monogenic disorder. So I wanted to include this quote from a paper by McCusick in Science a number of years ago, talking about mechanisms underlying monogenic diseases, which, of course, Barth syndrome is an example of. And as they say, there are major problems associated with understanding the pathology of even a simple monogenic diseases. And among the many reasons for this is the fact that they're modifying effects of other genes. And so clearly, until we understand the modifying effects, then it will be very hard to understand the pathology and also the relationship of genotype to phenotype. And of the 1,500 or so monogenic diseases, there's only a few where that relationship is really known. So I think it's important to keep that in mind. And based on that, we're very interested in understanding what are the functions of cardiolipin and what genes or pathways modify the cardiolipin deficiency. And so that's really been the focus of my lab currently. So in my lab, we use the yeast model system. And yeast is a very powerful model system because, for a number of reasons, the pathway, as Steve just pointed out, is well elucidated. The genes have been identified, and there are yeast mutants available for not only all of the cardiolipin genes, all of the genes in the cardiolipin pathway, but also all of the there are knockout mutations in essentially all of the other genes in yeast. This pathway is very highly conserved, and yeast are also amenable to genetic and genomic analyses, so you can get a lot more bang for the buck doing genetic analyses in yeast. The work I'm going to talk about today focuses on a mutant in this step, CRD1, cardiolipin synthase. And so this mutant this gene is responsible for synthesizing cardiolipin. So the CRD1 mutant has no cardiolipin, is blocked here. And it has the most severe phenotype. So if you look at these parameters that we just talked about, decreased CL, increased monolyso-CL, decreased fatty acyl species, this has no CL, so it has the most severe possible phenotype. And I mentioned that we have been interested in, and we have discovered that cardiolipin plays a role in many functions, mitochondrial, and also functions that are not simply associated with mitochondrial bioenergetics. We've talked about these over the years. And what I'd like to talk about today is the role of cardiolipin in iron homeostasis. So this study is, everything I'm going to talk about today primarily was done by Vinay Patel, who's here in the audience, and he has a poster. And it was initiated a number of years ago by a previous graduate student, Vishal Gohil, who did the microarray analysis that I'll talk about. Vishal is now an assistant professor at Texas A&M. I just saw him yesterday, and he actually looks grayer. After just one postdoc at Harvard, he's almost completely gray, but still the same Vishal. So this is the microarray analysis data that Vishal did, and he noticed that among all the genes that were affected in the CRD1 mutant, there was an increase in expression of genes in the iron regulon, the genes that affect iron utilization. And so these genes were upregulated. So what is iron? The iron regulon, iron homeostasis. It's a very, very complicated general pathway, as you can see here, in which there are siderophores that bind to the ferric form of iron. There are reductases that reduce the +3 form to the +2. There are permeases that transport iron at various stages into the cell and into and out of the vacuole and the mitochondrion. And these genes are many of these genes are coordinately regulated. So Vinay did the PCR analysis to validate the microarray, and he determined that sure enough, in CRD1, all of these genes show increased expression. So this led to the question, does the loss of cardiolipin lead to defective iron homeostasis? And there are three parameters that you would expect to see if there was a defect in iron homeostasis: perturbation of mitochondrial iron, decreased growth of the CRD1 mutant on media with high iron, it would be sensitive to iron, and also increased sensitivity to agents that induce ROS. So to look at the first parameter, does CRD1 have altered iron levels? And so as you can see here, we're looking at mitochondrial iron. There is an increase, it's a small but very significant increase in mitochondrial iron in the CRD1 mutant. And the second parameter, sensitivity to iron. So here we're simply looking at growth of the CRD1 mutant versus the wild type on plates with containing different amounts of iron. And you can see that the CRD1 mutant shows altered sensitivity when compared to the wild type and is hypersensitive to increased concentrations of iron. And the third parameter is regards to sensitivity to ROS-inducing agent. We can see that CRD1 is also sensitive to hydrogen peroxide. So if you plate E cells on plates that have increasing amounts of H2O2, you can see a greater sensitivity than is seen in the wild type. So all of these parameters have been found, and they suggest that the loss of cardiolipin leads to a defect in iron homeostasis. So how can we connect that? What is the relationship between this lipid in the mitochondrial membrane and increased expression of the iron regulon? Well, there are two sets of factors that cause increased expression of the iron regulon. One is if you starve cells for iron. If you starve cells for iron, they're going to want to increase the iron regulon to bring iron into the cell. But that's not the case here because the cells are grown in iron, in the presence of iron, so there's no starvation. So the other is defect in the synthesis of iron-sulfur clusters. Iron-sulfur clusters are, as you would imagine, clusters of iron and sulfur. And they are shown here. The major iron-sulfur cluster has 4 irons and 4 sulfurs, and there's interconversion to a form of 2 and 2 and 3 and 4. But this iron-sulfur cluster, the iron-sulfur clusters are essential cofactors in energy metabolism and in actually many reactions in the cell. So in energy metabolism, in the electron transport complexes, many of the enzymes require iron-sulfur clusters. And in the TCA cycle, the enzyme aconitase is also an iron-sulfur containing enzyme. So iron-sulfur cluster biogenesis is a very complicated process. And this is actually a slide from Roland Lill. You can't see that, but he is one of the foremost authorities on iron-sulfur biogenesis. And there are many genes that are involved in iron-sulfur cluster synthesis, as you can see here, but you can divide the process into 2 basic steps. One is the synthesis of the iron-sulfur scaffold, that's seen here. And the second step is transferring the iron-sulfur scaffold to the APO enzyme, which then becomes the holoenzyme that now has iron-sulfur. So we predicted that that the defect in iron homeostasis stemmed from a defect in some aspect of this process. Now, iron-sulfur clusters are also required for cytoplasmic enzymes, and factors come out of the mitochondria into the cytoplasm in order for cytoplasmic iron sulfurs to be synthesized. And for that to happen, you need this protein ATM1. And interestingly, again, this is from Roland Lill's lab, interestingly, cardiolipin stimulates activity of ATM1. So the natural question is, maybe the defect is because of not having enough ATM1 activity. Now, if that's correct, then you would predict that if you add more ATM1 back to the cells, then you would alleviate the defect. And so Vinay did that experiment. He overexpressed ATM1 in the CRD1 mutant, and that's seen here. So here we're looking at the fold of iron regulon gene expression. Here's the wild type, and here's the CRD1 mutant. And here is in the presence of the overexpression of ATM1. And so you can see that CRD1 has increased expression of these 3 genes that he looked at. And if you add ATM1, it doesn't decrease that. It doesn't rescue that increased amount of iron regulon and bring it back to wild type. So we can say that overexpression of ATM1 does not restore the wild type iron regulon. And so we're back to the hypothesis that perturbation of iron homeostasis may affect iron-sulfur cluster biogenesis. Now, if that's correct, then we can make 3 predictions. One, that the cardiolipin mutant would exhibit decreased activity of FES enzymes, right? Because if you're not making enough FES, then the enzymes that require FES are going to be reduced in activity. And 2, that the CRD1 mutant would be sensitive to further perturbation of FES synthesis. If FES is already decreased, then if you decrease it more, you're going to make the cell sicker. And 3rd, that CRD1 would exhibit upregulation of the FES scaffold, because this is what is seen characteristically in cases where FES is perturbed. So we look at this first, activity of FES enzymes. And here we can see activity of mitochondrial FES enzymes. Here, Vinay looked at succinate dehydrogenase, ubiquinol cytochrome C oxidoreductase, and aconitase. And you can see that all of these enzymes in CRD1 are decreased in activity relative to the wild type. So in the case of mitochondrial FES enzymes, activity is reduced. And Vinay also looked at cytoplasmic FES enzymes, isopropyl malate isomerase and sulfite reductase. And these two are also decreased about 50% relative to wild-type cells. So both mitochondrial and cytoplasmic FES enzymes are decreased. Now, these cytoplasmic enzymes that I just talked about are involved in amino acid synthesis. So if the activities are decreased, you would expect to see that the cell would be oxytrophic or would require specific amino acids. And sulfite reductase deficiency would be expected to lead to a methionine oxytrophy. And isopropyl malate isomerase would be expected to lead to a leucine oxytrophy. And that's what we see. So here the CRD1 is plated on cells with and without methionine. And you can see that at the elevated temperature, it doesn't grow without methionine. And similarly, when the cells are plated on plates with and without leucine, the CRD1 mutant cannot grow at elevated temperature on plates without leucine. So we can say that the CRD1 does in fact exhibit decreased activity of iron sulfur enzymes, and that this results in a phenotype of requiring amino acids. So now let's look at the second parameter. We would predict that CRD1 is sensitive to further perturbation of FES synthesis, or as we say in the genetic lingo, exhibits synthetic genetic interactions. If two components participate in a similar pathway, if you knock down one, then the cell will be more sensitive to knocking down the other. So back to the iron-sulfur biogenesis pathway. So here, again, you see the synthesis of the iron-sulfur cluster. And essentially what happens is that all of these genes participate in bringing iron into the cell, in liberating sulfur from cysteine, in delivering it to the scaffold protein ISU. And so the question is the cardiolipin mutant synthetically lethal with any of these mutants? But you can't really look at most of these because these are essential genes. Most of these are essential genes, all except for ISU, because ISU, there are two genes that encode for ISU. So you can look for synthetic interaction between CRD1 and one of the ISUs, which is the scaffold for the iron-sulfur cluster. And in this experiment, here you see growth of the wild type and growth of the CRD1 mutant and growth of ISU1. And what happens when you make the double mutant, CRD1, ISU1, you can see a synthetic interaction. There's decreased growth, suggesting that the ISU1 mutant exacerbates the growth defect of CRD1. So that says that CRD1 is sensitive to further perturbation of FES synthesis and further supports a role for cardiolipin in FES biogenesis. And now let's look at the third parameter. There are several papers in the literature that show that if you have perturbation of iron-sulfur synthesis, you see upregulation of the FES scaffold. And these are papers from Elizabeth Craig's lab. So we look to see whether ISU levels are altered in the cardiolipin mutant. And that's shown here. Here we can see mRNA levels, and here we see protein levels. And you can see that very clearly ISU is elevated in CRD1, ISU protein, and also ISU mRNA. So this is consistent with the third parameter that CRD1 does exhibit upregulation of the iron-sulfur scaffold, consistent with an FES defect. So to summarize these data, we can say that cardiolipin leads to an iron homeostasis defect and also perturbation of iron-sulfur biogenesis. We see increased mitochondrial iron, sensitivity to ROS-inducing agent. All of these are phenotypes of iron homeostasis defect. And FES-associated defects are decreased activities of mitochondrial and cytoplasmic FES enzymes and synthetic interaction with iron-sulfur scaffold protein mutant and other phenotypes associated with an FES defect. So we propose this model where you have a decrease in cardiolipin leading to a decrease in FES biogenesis, causing a decrease in activities of these FES proteins. And this is detected by the transcriptional activator AFT1, which goes into the nucleus and increases expression of the genes in the iron regulon. And this causes increased iron uptake into the cells and into the mitochondria. That's it, which would result in the increased mitochondrial iron levels. So that's the model. Now, how does this all happen? What's the mechanism linking cardiolipin to the FES defect? So we hypothesize, based on studies that we have published previously, that cardiolipin is required for optimal mitochondrial protein import. We suggest that the likely mechanism linking cardiolipin to defective iron homeostasis is defective import of proteins that are involved in FES biogenesis. This is kind of a modification of Richard's hypothesis, but not totally inconsistent with it. So, what are the consequences of iron-sulfur defects, and how might that be related to Barth syndrome? And here again is echoes of Richard's talk. So here's the TCA cycle again, which has popped up before. And I mentioned that we have seen a decrease in aconitase, the FES mitochondrial enzyme aconitase, and succinate dehydrogenase. And these are TCA cycle enzymes. So if there's a decrease in TCA cycle enzymes, you expect to see perturbation of the TCA cycle. That would be expected to have major secondary metabolic effects, because just looking at amino acids, as Richard has talked about and Todd, just looking at amino acids, these are places in the TCA cycle where amino acids feed in. So now you're going to be tested on which amino acid goes where. But if we just look at this step right here, alpha-ketoglutarate is important for the synthesis of glutamate. And glutamate is required for synthesis of glutathione, which is a very important antioxidant. Glutathione is actually a tripeptide consisting of glutamate, cysteine, and glycine. And so if alpha-ketoglutarate is deficient, then you would expect glutamate to be deficient. And I mentioned earlier that sulfite reductase is defective because that's an FES protein, and that plays a role in the synthesis of cysteine. So you would expect to see a decrease in glutathione synthesis. And if that's the case, then supplementing with glutathione would rescue some of the phenotypes. And so here's an experiment that Vinay did showing that if you supplement glutathione, you can restore the growth defect of the CRD1 mutant. Here's a CRD1 mutant without supplement, and here is in the presence of glutathione. Now, Vinay told me I should make sure to add this next slide, so this is for you, Vinay. If you supplement with glutamate, you also restore growth of CRD1 at elevated temperature. Here's CRD1 unsupplemented and supplemented with glutamate. So it fits nicely into this story. Now, Richard very kindly provided me with his data that he talked about, but I want to show here because it's very relevant. You may not expect to see the exact same differences between that you see in yeast in humans, but you would expect to see perturbation of the cycle. And so Richard has shown that there is a huge increase in aconitate and isocitrate in Barth patients, and also in amino acids, most notably a decrease in arginine. So our findings, I think, offer a possible mechanistic background in which to understand how these defects may occur. I think I'll just skip this. So we suggest that the loss of cardiolipin leads to defective iron-sulfur biogenesis, which causes perturbation of the TCA cycle, and subsequently many secondary metabolic dysfunctions. And what we're hoping to do next is to pinpoint the mechanism linking cardiolipin to FES biogenesis and to determine the consequences of TCA cycle perturbation in cardiolipin deficient cells. And I didn't talk about this, but what's also linked, and actually Vaishnavi Raja, who is here, a student in my lab, is talking about the role of cardiolipin in acetyl-CoA synthesis and beta oxidation, which of course will affect the TCA cycle. So I mentioned that Vinay did virtually all the work shown here, and he's just about to graduate, so looking for a postdoc. He's really good, so if anybody has a postdoc for him, I recommend him. Vishal did the initial microarray, and our collaborators at University of Utah, Dennis Wenge and Jennifer Fox, helped us with the iron assays, and we acknowledge support from NIH and much support from the Barth Syndrome Foundation, for which I am very grateful. Thank you very much.
Great talk, Miriam. Questions?
Thank you.
Roger?
The obvious question would be whether this kind of defect happen in your tafazin mutant, the iron sulfur. And a related comment is that, the red blood cell does not have mitochondria, and red blood cell has a lot of iron. Whether this has any correlation with your finding or not, I would like to hear your comment.
Well, the question about whether the tafazin mutant has defects, that's a really good question, and it's a very it's very complicated to answer that because what Vinay has seen is that some of the defects are seen in the tafazin mutant and some are not. And in the cases where we do see the defects, of course, there's a more severe defect in CRD1 and then a less severe defect in TAS1. You want to add anything to that? So I would say that TAS1 shows an intermediate defect, maybe because it has some cardiolipin, some is better than none. I think that's probably a fair statement, that if you lose cardiolipin entirely, you're going to be in much worse shape.
Miriam, very nice work. I was wondering, in order to reconcile your data with Richard, I still have a difficulty actually, because if aconitase is impaired because it has an iron sulfur center, you would suspect that citrate would be high, and aconitate would be lower, and isocitrate would be lower. And with Richard, it's the other way around. Citrate is normal, aconitate is higher, isocitrate is higher. So how do you see that?
Well, again, I don't expect the exact defects to be the same, but just and we of course don't know the nature of iron sulfur perturbation, if there is any in Barth cells. This is something we're going to look into, but I think you'd expect to see lots of defects, even though the underlying mechanism may be the same. So I can't really answer that specifically until we really look into the iron sulfur metabolism in Barth cells.
Beautiful work, as always. Have you showed that like aconitase and some of these enzyme activities were low? Since another possibility is defective import that could lead to reduced activities, have you guys correlated the protein expression levels from the delta CRD to wild type and the activities? Have you been able to?
I leave that to you, sir.
So we looked at succinate dehydrogenase and ubiquinol, the complex 3, and those are not altered. Aconitase we have not looked.
But if you look at steady state levels, may very likely give you a different answer than looking at the kinetics of uptake.
Anyone? Okay. Yes, I agree with Ron. However, there's something I just thought of. One of the first abnormalities we found in Barth syndrome was increased aconitate in the urine. And we often see increased levels of citric acid cyclic intermediates in the urine in mitochondrial problems, and it's always been a puzzle, or not a puzzle, but it's been a question whether it's just the kidney or a systemic abnormality. And one of our findings in doing the plasma citric acid cyclic intermediates is that we can have a 20-fold increase in a citric acid cyclic intermediate in the blood and have a completely normal level in the urine. They're valuable substrates that are reabsorbed by the kidney. Therefore, and again, when we measure in the blood, we're measuring largely what's going on in the liver and some in the muscle. In mitochondrial problems, they're typically systemic, so that's a reasonable way of approaching things. But then it does raise a question in view of your hypothesis. In the kidney, could it be that I'm sure that what we see is only sort of the tip of the iceberg. In the kidney, could it be shifted somewhat so that the iron sulfur problem is more prominent? The other thing is that there is, I'll leave it right there. That's one. It's an interesting possibility that I have to look upon this old finding of high aconitate in the urine as being more important in terms of what's going on in the kidney.
Well, this raises again Michael's question earlier this morning, that to what extent do the plasma levels reflect what's going on inside the cell? So, we hope to have those answers by going to Michael's lab and doing some collaborative work with him.
Any other questions? Okay, if not, we'll get to the